All right, we have a quick little video here to talk about the ways that gene expression in prokaryotes is similar and ways that gene expression in prokaryotes and eukaryotes is different. If you'll remember, gene expression is just taking the code of DNA and expressing it into something the cell can use, whether that's an RNA or a protein. And so one big difference is that prokaryotes don't have a nucleus. So transcription normally happens. Occurs in cytoplasm. Normally, or I should say for us, transcription occurs in the nucleus because that's where the DNA is and it can't leave the nucleus. It's a prisoner in the nucleus. So transcription has to happen there. But they don't have a nucleus, so their DNA is just floating around the cytoplasm doing its thing. Because I should say DNA is in cytoplasm. It's not really a difference in gene expression, but it's a difference in the way that everything is located, the geography of the cell, so to speak. All right. Another difference is that DNA polymerase, I'm kidding, RNA polymerase. Going back to replication. We don't want to go back to replication. RNA polymerase binds to the promoter, just directly binds. Nothing else has to happen. It's all good. Whereas in eukaryotes, transcription factors, I'm going to abbreviate those, transcription factors bind to the TATA-box, which is just a series of, I think it's TATAAAA. Those transcription factors bind to the TATA-box. Then RNA polymerase can bind to the promoter. So there's just an additional step that eukaryotes have to do. It gives us a little more control. In this one, there's no mRNA processing. And so when that mRNA is made, and no introns, which is part of that, but when that mRNA is made by transcription from the DNA, it can go directly into translation. And actually, a lot of times, the ribosomes will go ahead and bind to the mRNA before it's even finished being made. So, and this has mRNA processing. And remember, that processing is that adding that G5' cap, the poly A tail on the 3' end, and taking out those introns. Removes introns. So how are they similar? Well, they both do transcription and translation. They both have all the RNAs, so mRNA, tRNA, rRNA, which rRNA is part of the ribosome. Now, the ribosomes are different, but they do both have ribosomes. It's the one organelle that prokaryotes do have. And that's probably the main, I'm trying to think through. They both use RNA polymerase, a lot of the same enzymes or versions of enzymes. It might not be exactly the same, but they both use RNA polymerase. They both do A to U, G to C, and the code is universal. So if we're talking about that codon chart or that codon wheel, it's the same for both. So those are some ways that they're similar, as well as some ways that they are different in the way that they do gene expression. And I hope that helps.